would like to do is to also show you the fact that mouse models, and again, what I'm trying to do is not tell you only about what we do in the lab, but what other people are doing. Telling you that mouse model where people have activated an oncogene and mutated the tumor suppressor gene have been instrumental in modeling many cancers, such as colon cancer, prostate cancer, breast cancer, ovarian cancer, etc. And there are resources on the website. If you go and look for the mouse repository, and you can Google this, and you'll find different mouse strains which you actually can see for that characteristic and how close to human they are in terms of their disease presentation. And you'll see they'll also tell you about their phenotype and how they present with disease. This is a great resource. And again, this is an example of all the mouse strains that have been generated and which are on this repository. And there is also another repository which is in the MMRC, which you can look up as well. And there is the MGI, which is also a great resource. I think this was developed initially by JAX, where you can look for gene expression, gene mutation, etc. So I think in summary, I told you about what we were doing in the lab. I'm also showing you that mouse models are a great tool for studying human disease, although they have limitations and one has to be very careful, and you cannot translate what you see in mice data to what you see in human, but you have to just translate the concept you learn from that. This is our group, and everybody who helped in doing what we do. I think everybody who has participated in this, including Andre here, Eunice, who has been in the lab and is in the audience, Bilal, who has just joined, and everybody else. Alan Houghton, who has created this program, and Jen Walker has been doing it, all our collaborators, and thank you for your attention. Any questions? Yes?
Can you turn back to the chart with the target there, positives and negatives, and the immunotherapy?
This one?
Okay.
So that is not a question. Okay. Yes? Oh, can I talk more about this? So basically the take-home message is that the targeted therapies are small molecules, okay? So what you do, you look at the patient's tumor and you try to determine what mutation or what change they have. And once you know what change they have, you go on your shelf and you pick the drug that corresponds to that mutated gene. Okay, and then, so when you give them that, then you are blocking and the mutated gene is an oncogene. So you are blocking what causes cancer. Okay, so, and then those cancer cells will die. Okay, when these cancer cells will die, they will be giving antigen, or they will be giving protein to the immune system. That's what we think. So it will be like immunizing your immune system against your cancer cells. Okay, but then when you give the immunotherapy, which is a simple way of saying it, is blocking the brain or stopping whatever says to the immune system do not kill. What happens is that the immune system will be unleashed now, and you just can't kill. But the fact that you were able to kill cancer cells already with the drug, it's like telling the immune system, this is what you can recognize, and you can go after this, that protein. Is that clear? Okay. Yeah. Very good.
I understand that melanoma is skin cancer, but how does it affect our inner organs?
So the reason it affects, yes, I'll tell you how it affects your inner organs. So if you remember, the first Okay, here. So if you remember this slide, what happens is that these melanoma cells, which are cancer cells, they have accumulated changes, and those changes allows them to migrate. Okay, so they are going to go to the blood vessel or the lymph, and now they can move in your body. Okay, and they lost all the information that tells them just stay here and don't move. Okay, so now they are going to migrate, and wherever they go, they will stay. Okay, so if they go to the brain, then they'll stick in the brain, and they'll start dividing, and then you'll have a brain tumor. If they migrate to the lung, then they'll start dividing in the lung, and you'll have a melanoma in the lung. This is called metastasis. Does that answer your question?
Yeah, it begins on skin and then it travels.
Yes, it begins in the skin initially, and that the form that is in the skin doesn't travel anywhere, right? But then when you get more mutation or more changes in your DNA, then they acquire the ability to travel and go somewhere else. Okay?
Two questions. For immunotherapy, does the receptor protein change over time? And the second question is how long does it last? Immunotherapy, is it 1 year, 10 years?
So the first question is both possibility. So it is possible that it changes. It's possible that it doesn't change. Okay, and that is something that will determine how long the immunotherapy will last. And that's what we call immune escape. Okay, so the tumor cells are going to escape your immune system and will come back. And so there are patients which have been treated at Memorial Sloan Kettering Cancer Center and have remained free of disease for 7 years. There are other patients who, had disease after 1 year. So it's very variable, and the reason why we are doing research is we don't understand everything yet, and our goal is to have it last as long as we can. I had a question there. Go ahead.
I'd like to ask, why does melanoma why the treatment with radiotherapy and chemotherapy, as we were saying before, isn't as useful, isn't as helpful as targeted therapy and immunotherapy? And also,
that's fine. I'll answer this question, and then I'll come back to you. I'll answer somebody else. So the thing is, first of all, we don't know. So chemotherapy and radiotherapy are treatments which work very well or quite well for most cancers. Okay, chemotherapy will kill any cell that's dividing because you are just giving something toxic, and so we'll preferably kill whatever is dividing. I think radiotherapy is the same idea, but the melanoma are resistant because the pathway which we turn on make them resistant to something called apoptosis, which is induced by chemotherapy or radiotherapy. And one of the reasons why immunotherapy has been investigated so much for melanoma, and we know it's starting to work, is because it was not working for radiotherapy and chemotherapy were not working in melanoma. That's why people started working on it much more, and also because it's very easy to detect, and there are things which are called differentiation antigen, or the things which make melanin, have been very widely studied, so it made this as a model of choice to study immunity in melanoma. And the targeted therapy do work in other diseases as well. It's just that now, so these targeted therapies have been through many reiterations, so they tried many small molecules which do block these mutations in the cancers, and before they are not working, but now they're getting to second, third generation, which are working pretty well.